Welcome everybody. Welcome to SERCAL 2020, Shedding Light in Challenging Times. I'm Thor Anderson. I'm your conference chair, and this is SERCAL's 27th annual conference and our first virtual conference. I want to make sure you all could hear and see me. Maybe someone could send a chat out letting me know that I'm live. Thank you. Just seeing the chats real quick. Good, I'm live, everyone can see me, great. So today's Thursday, September 3rd. This is our second session. We kicked off the conference on Tuesday with a plenary speech from Bruce Delgado, which was great. And he also chaired the Managing Invasive Species session that went really well. And today we're moving on to grassland ecosystems. This is part one of a two-part session. So the second grassland ecosystems will occur on September 17th. So I hope you can make both of these, and I hope you can make all 8 of our sessions throughout the month. I want to give a big shout out to all our session chairs. They've been great. They've been volunteering their time and been very thoughtful at putting together their sessions. A big shout out to Bruce Delgado for his plenary speech, which was fantastic on Tuesday. He addressed the Black Lives Matters issue, which is really important to address. And then he talked about all the great collaboration and creativity going on at Fort Ord National Monument with restoration and the management of those lands. So I found that really inspiring. If you missed it, you could, we're recording all these webinars, so you could catch that, and we'll be sending out an email with links to watch anything you missed, or if anything you wanna watch again. So thank you all session chairs. And big thanks to all our Golden Opportunity sponsors. We have a long list of sponsors this year, so a lot of groups are continuing sponsoring with us, and we have some new ones. So thank you very much. Shouts out to HT Harvey and Associates, SWICA, SNS Seeds, ICF, Balance Hydrologics, Burleson Consulting, Westervelt, Dudek, HRS, Wildlands, Marin Municipal Water District, and Rocky Mountain Bioproducts. Thank you for helping us put this conference on. Also want to give a shout out to our silver lining sponsors with Cal IPSI, the Watershed Nursery, NHC, WRA, CBEC, UCI, and California Native Grassland Association. And today, the president of California Native Grassland Association, JP Marie, is our session chair. So thank you, CNGA and JP. Also, if you didn't get enough of Managing Invasive Plants session from Tuesday, Cal IPSI is putting their conference on in October. October 27th through 30th is their conference. So if you want to attend that, that's great. If you do SERCAL and Cal IPSI conferences, there's a discount for you. So look into that. Want to give a shout out to any of the individuals and firms that sponsored a student. We have 21 student sponsorships so far, and we had 23 students attending Tuesday's session, so great turnout. And I think we have a total of 30 students who are registered for the conference. And I do believe now we have over 200 people registered for the conference. And what do we got? It looks like we have 91 people attending right now, and the number keeps kind of trickling up. Today's session is, we have special sponsors for each afternoon session, and Dudek and HRS have joined us for that. And we have Cindy Thompson and Kyle Matthews. If you guys want to join the virtual stage here and tell us a little bit about the services that HRS and Dudek provides.
Yeah, thanks, Thor. Appreciate you guys giving us the opportunity to speak. Yeah, so HRS and Dudek are really full service implementation and design, permitting, everything top to bottom for all your environmental and engineering service needs. So DUDEC handles, permitting, compliance, biology. You see all the services listed there. And then, I personally am on the HRS team, and so we're the boots on the ground implementation construction arm. So we really like doing habitat restoration, and that can range from grassland ecosystems like you're going to hear about today to, river and creek restoration, montane meadows, coastal sage scrub, the whole slew of it. So thank you.
Great, thanks, Kyle. And Kyle and Cindy and other HRS and DUDEC staff will be in the meet and greet room that's going to happen at the end of today's session. So don't log off if you want to do some meeting and greeting with them and ask Kyle any question you want. So don't log off. We will invite you to share your video and audio so we could go into a Zoom-like meet and greet room. We also have another special session sponsor today with S&S Seeds. So Jody Miller will be in the meet and greet also this afternoon. So if you have questions for Jody, come join us. S&S Seeds is a commercial seed supplier. They specialize in seed production on their farm. They can collect wildland seed. They have storage and processing facilities for seed. They can help you with design. They also could provide you with erosion control products. They are located in the Santa Barbara area with an office and warehouse in Carpinteria, and their production fields in Los Alamos. They have 450-acre and production fields, about 120 fields ranging from a 1/4 acre to 20 acres in size. And they have specialized equipment to produce that native seed for restoration projects. They could also help you with seed mix design and explain the difference between bulk seed and pure live seed, and help you get the appropriate amount of live seed on your restoration areas. So they also have seed you could purchase of approximately 1,000 species, and they could work out agreements with you either to collect site-specific seed or to contract grow to produce seed. So if you have questions, Jody will be with us in the meet and greet this afternoon. So thank you, S&S Seed. Here's my last slide before we start our session today. Just a few rules and reminders and recommendations of the webinar here. So panelists, please, hide and mute your video and audio as appropriate. So just pop up on the screen when you're presenting. Attendees, you're all automatically muted and hidden. However, when we go to the meet and greet session at the end of today, we will invite you to share your audio and video. So please share your video when you come to meet and greet. The chat feature is an optional feature. It's a real fun way to connect with everybody. So if you use it, please be respectful and treat others how you would like to be treated. And if you have formal questions, please use the Q&A feature. So that will be formal questions for the presenters, and they'll do their best to answer all your questions at the end of their presentations. And finally, we're recording all these, so you'll get plenty of opportunities to watch again if needed. So we'll be sending out emails in the future with a link to the recordings. All right, let's get to today's session, Grassland Ecosystems. Our session chair is JP Marie. He's the president of the California Native Grassland Association and also represents UC Davis. JP has been supporting SERCAL for years. He's been session chair for Grassland Ecosystems sessions many times. It's a great one. I always catch it when I come to the conferences. He always has a great group of presenters and shares, has so much to share. So he's here again. Thank you, JP, and welcome.
Thank you, Thor. Can you hear me okay?
Yes, I could hear you fine.
I was going to turn my video on, but it's not able, but that's okay. Okay, so I'm going to give a quick introduction. I want to thank Thor and Julie and the entire SERCAL board for hosting a Grassland session, part of the conference. I'm really excited about this. I want to thank all of our sponsors, specifically for today, HRS, DUDEC, SNS Seeds. I'm really looking forward to some exciting talks. As far as logistics go, today is part 1. It's going to be a little shorter. We have 5 presenters, including a lightning talk. So plan on being here for about 90 minutes. On the 17th, we have 7 full presentations. So on that day, plan on being here for about 2.5 hours. Today, I would like to, sorry, I'm a little emotional. I would like to dedicate our Grassland session to John Anderson. Some of you might already know, but John passed away a couple of weeks ago. You know, he was one of the founders of CNGA, California Native Grassland Association, founder and owner of Hedgerow Farms. I could probably spend an entire week talking about John. He was involved in many boards, conservation-oriented board throughout his career, including the National Audubon Society, Audubon California, Yellow Basin Foundation, Wild Farm Alliance, and the Yellow Resource Conservation District. He received many awards during his career. The last 2 I remember was for Calypsy. In 2008, he received the Ryan Jones Catalyst Award, and in 2014, the Jake Sieg Award for Vision and Dedicated Service. And, you know, he's always moved by awards, but he always say he doesn't want awards. That's not why he does what he does, sorry. But I think the one he got the most moved by was in 2017, he received the SERCAL's first ever Lifetime Achievement Award. That really meant a lot to him. So thank you, SERCAL, for awarding him that award. So again, we're dedicating an entire Grassland session to John Anderson. He will be greatly missed. All right, so today we have 5 presenters. Taraneh Emam, who's going to do a lightning talk on translocating mature needlegrass for restoration. Sarah Gaffney, who's going to talk about native grass resistance to noxious weeds. Christopher Hart, goat grazing on coastal grasslands. Julia Michaels, positive impact of grazing or vernal pools. And we'll end with Brianne Palmer, who's going to do an annual update on BioCrust. So, Taraneh, you're up. Taraneh, you can share your screen if you like. Taraneh Emam is a senior biologist and regulatory project manager at eCorp. She has a background in restoration ecology, primarily working in California grasslands. At eCorp, Taraneh manages permitting per state and federal regulations for a variety of project types, specializing in mitigation of project impact through preserve establishment and restoration. Taraneh also leads an eCorp team which tracks and analyzes changes to environmental regulation. Taraneh, thank you very much.
Hi, JP. Is my screen sharing working okay? It says that
It is
not able to start video. Is that all right?
Yes.
Okay. All right. Well, I'm going to give a brief overview on needlegrass translocation today. It's not going to be, there we go. Okay, you should be able to see me now. So yeah, I'm going to give just a brief lightning talk on needlegrass translocation. It's not going to be super academic. It's mostly going to be anecdotal information in the hopes that it'll be useful to some of you in some of your own projects. So the title of my talk is Needlegrass Translocation, It Works, and you'll note the asterisk that I added on the end. So the rest of the presentation is providing all the caveats to this statement. The brief background is that I've been working on a development project in the Sacramento region that had a CEQA mitigation measure requiring translocation or transplanting of existing needlegrass plants in the field that would have otherwise been graded over. I was initially fairly skeptical that this method could be successful because bunch grasses invest so much of their biomass in roots below ground, and a lot of that would be lost when translocating mature plants in the field. So I tried to look for evidence in the literature that this could be an effective method, and I didn't find that much on the topic. So of course I called John Anderson, and he told me that he had had success with translocating needlegrass. So that was very encouraging, and we decided to give it a try. So basically I'm just going to share my thought process and approach to successfully transplanting needlegrass. One important thing I'm leaving out is site selection, because that's not very specific to translocation as a method, but feel free to ask me about that later if you like. All right. So the most important thing to understand when translocating needlegrass is the phenology of bunch grasses in our climate. I didn't do any intentional experimenting with translocating at different times, but the phenology gives us some guidance as to the best time to translocate. As I'm sure you know, the growth season for perennial bunch grasses starts at the beginning of the rainy season, usually late fall or early winter in the Central Valley, and that's when plants start to move or reallocate biomass that's stored underground in their roots to greening up above ground and growing new shoots. While growing through the wet season, the resources are being allocated to both shoot and root growth. And then in the spring, it starts to be allocated more towards reproduction, so producing flowers and seeds, and then primarily back to roots again in preparation for summer dormancy. So in theory, you would want to transplant bunch grasses not first thing in the start of the rainy season, but once they've had a chance to reallocate biomass from below ground to above ground, because otherwise you're going to lose a lot of the root mass when transplanting. Now, despite understanding the phenology, sometimes there are logistical issues that get in the way. The first year of translocating, the schedule kept getting pushed back to reasons beyond our control, so we ended up attempting to transplant in April. The plants weren't flowering yet, and we were having late rains at that point, but of course, as soon as we translocated the plants, the rain stopped and the weather got very hot. And on top of that, we had some complications with getting them watered for the first few weeks due to some logistical issues. So needless to say, there is some high mortality during this first round of translocating. And I did notice anecdotally that the larger plants and the plants that weren't as close to flowering at the time that they were transplanted had higher survival. So that was a good clue to which plants were going to be good candidates for translocating in the future. So then we did 3 additional translocations in the following years, and things went much more smoothly for those. So all those plants got transplanted in February and March. And when there were breaks in rain during the winter, where there was more than a couple of weeks of a dry period, we had them watered by SMP, our restoration subcontractor. For all the transplants, we watered them every 1 to 2 months over the summer just to get them through their first dry season. So that was just for the first year. And they're all doing pretty well now from these last 3 translocations. We did find, though, that the management of annual grasses was really important, as it is in any native grassland restoration. So before each translocation, the only weed management that was done was 1 round of mowing of the site before the plants were transplanted in. So obviously the annual grasses bounce back quickly, and we found that by the 2nd year after transplanting, weed management was needed to keep the needlegrass happy, particularly because there was a lot of medusa head at our site and the thatch doesn't break down as quickly. So for our site, we did this by mowing and raking up the thatch and removing it, but it could also be done by grazing for a larger area. So now I'm going to go through and show some photos of the translocation that we did in 2018, since it's now had a couple of years of monitoring. So this round was transplanted in February. You can see that the ground was hard enough that SMP had to use jackhammers to get the roots out of the ground. We tried to get about 10 to 12 inches deep to get a nice root mass. Needlegrasses and bunch grasses in general can have roots several feet deep, but the majority of the root mass is in the top foot or so of the soil. So that's what we aimed for. We tried to keep the root ball intact with minimal disturbance of the soil. As I mentioned, they were watered every 1 to 2 months over the summer. And in September, there was a lot of senescence, but you can tell that they are still doing okay because they had leaves that were green at the base, and that's what you want to look for to make sure that they're surviving and still healthy, as well as the crown still being intact and not being brittle when you pull on it. So after 1 year of growth post-transplanting, they were still looking good, and we had a 92% survival rate over the first year, which was pretty great, better than I expected. And in the first year, there wasn't a lot of thatch buildup yet. You can see there's a lot of annual grasses growing, but not a lot of buildup yet. And then after 2 years post-translocating, there was a lot of Medusa thatch building back up. So the first photo is from February, and all those kind of tussocks you see is where the Medusa head thatch is actually, building up on top of the needlegrass bunches. So starting to shade out the needlegrass. So at this point, we weed whack and bagged up the thatch and removed it. And the second photo is post-mowing in May, and so they're looking a lot happier now. So the take-home message is that needlegrass translocation can be really useful when the conditions are right. For example, when there's an existing population that would otherwise be impacted by development and you can salvage it. It's probably not well suited for large areas because it is very labor intensive to dig up the plants and to dig larger planting holes than you would for plugs. It might be useful when you aren't planning to do a lot of site preparation to get rid of non-native plants, since these are large, mature plants that have already been growing in competition with non-natives in the field. Translocating can also help preserve the genetic diversity or ecotypic variation of these populations that have been doing well competing with non-natives and are adapted to that, as was the case in these translocations. And as you learned from our experience, translocating is really only worth it when it can be timed right and when you have water available to get them through at least just the first spring and summer through any hiccups or drought spells. And lastly, they of course will need long-term management to keep the annual grasses and thatch in check. But that's true of all native grassland restoration, as everybody knows. So that concludes my talk, and please feel free to ask me any questions or send me an email if we don't have time to get to everything. Thanks so much. And I will.
All right. Thank you, Taraneh.
You're welcome.
Let's see. So attendees, if you have questions, just go ahead and type in the Q&A. I do have some questions, but I'm going to go with the attendees' questions first. With regard to thatch management, what season is at best performed?
So that depends on, what the species of grass, of annual grass that are building up are. So for, your general oats and things that mature more quickly, I would say early spring, but you want to, it depends on the method too, because you want to avoid mowing. If you're going to mow, you want to avoid mowing the needlegrass when it's going to flower so that you're not impacting its seed production too much. So I think there's, a bunch of things that you want to juggle. You want to time the thatch management for when the annual grasses are at their kind of peak biomass and flowering, because that's when you're going to knock them back the most. But you also want to not impact the needlegrass as much as you can, especially when it's flowering and producing seeds. So depending on the suite of species at your site and what the climate is like, this, that might be easier or harder to juggle.
Excellent. For thatch management again, how much thatch did you remove? All or as much as you could, or maybe closer to 50%?
So SMP did the mowing for us, and they mowed it pretty close to the ground. So they, because they were using a weed whacker, they were able to kind of go around the needlegrass bunches and just remove the annual grasses that were growing in between. So they tried to remove as much as possible. I think it would be different if you were using a mower where you had to also kind of indiscriminately mow the needlegrass too. You would want to not whack it down too much.
Sure. All right. As far, I don't know if you looked at the cost involved into the translocation of the needlegrass.
Yeah.
How is it compared to planting from plugs or seeds?
You know, I think it depends on the factors that you have at your site. It's definitely really labor intensive to dig the needlegrass up. So, you know, and to dig larger planting holes. So, you know, compared to plugs, it definitely takes more work because the plugs you purchase from a nursery and you just have to make small holes to put them in the ground. But I think for sites where you're not investing in doing a lot of weed management before you plant, this could be a good option because they're better able to compete with the annual grasses that are growing there. So, you know, I think either your money is going to go towards knocking down weeds at a site before you plant plugs, because the plugs will have a harder time competing, or you know, can spend the money towards transplanting the existing needlegrass in a field, you know, when that opportunity is available, when there's existing plants that are available for salvage.
All right, thank you. There's a few more. Now, we won't be able to answer them all. So, Taraneh, before, if you have to go, whatever, just make sure you go to the Q&A and then you can answer the other ones on your own time.
Sure thing. Yeah, and feel free to email me too if anyone
we still have
wants.
time.
Yeah.
Did you use any soil inoculation on this project?
No, I didn't. And I don't know who asked that question, but you might be familiar with some of my previous work. I think all my past presentations at SERCAL have been on soil inoculation. So in previous research that I did for my dissertation, I found that, you know, using local soil where the native plants were growing turned out to be the best inoculant that most promoted native grasses, especially when it was targeted to the native grasses and not, you know, wide broadcast. So, you know, by transplanting an existing plant with all the soil intact around its roots, that's really, you know, the best inoculum in my opinion.
Very good. Okay, I'm going to ask you this one. Regarding transplanting of needlegrass, were measures taken to minimize the risk of spreading soil pathogens? Or any phytophthora
yeah.
testing?
So in this case, no, because we were basically taking things from an area that was going to be developed and transplanting just, you know, in some cases a few meters away, in some cases a mile away, but it was all, you know, within a pretty localized area. So I think, you know, that would be a concern in some other areas, or if you were doing translocation across a longer distance. But in this case, it wasn't really a concern for us.
Okay, that makes sense. Okay, I'm going to ask you two questions I'm going to combine in a single question. What was the time lapse between digging up the plants and replanting them? And what kind of method of irrigation did you use?
So the time lapse, we tried to do it basically as soon as possible. So we had, because it was a pretty short distance between the origin site and the translocation site, we were able to just dig them up, put them on the trailer of an ATV and drive them over as soon as the trailer was full. I think we kept them under a tarp when it got really hot and sprayed them down a little bit to make sure that they weren't getting too heat stressed. But yeah, I think the longest delay that we had was, you know, there might have been some that sat out overnight, but that was about it. I'm sorry, I forgot the second question already.
The type of irrigation.
Oh, type of irrigation. So that, because we weren't irrigating very often, it was just a hand pump and a creek nearby and then hand watering. We didn't have an irrigation system set up or anything because it was pretty infrequent.
Okay. All right, one more. How did you determine what the best planting density was for translocation?
So because the mitigation measure that we were working under was addressing valley needlegrass grassland as an ecotype and not needlegrass plants themselves, the focus was on the acreage of grassland. So, you know, within these grasslands, really, needlegrass was the only main native grass species, so that's what got transplanted. But basically, we took an acreage of needlegrass grassland that had, you know, a cover of 10% or more, and then basically transplanted into an area that was that same acreage, so the density ended up being the same.
Excellent. Thank you, Taraneh.
No.
So there's a few more questions, but we don't have time to answer them. But attendees, please keep typing your question on the Q&A. Taraneh is going to answer them behind the scene. And then later on, if you have any questions or whatnot for her, you see email address right there, teamam@ecorpconsulting.com. So Taraneh, thank you so much. All right. Our next presenter is Sarah Gaffney, and I forgot something. Thor did say I was the president of CNGA, but my main job, my paid job, I actually work for UC Davis, where I manage the Puta Creek Riparian Reserve, and my department allows me to be involved in those conferences and so forth. So I just want to acknowledge that, yeah, I work for UC Davis, and that's why I'm here too. Hello, Sarah. All right, so Sarah is going to talk to us about native grass resistance to noxious weeds. Sarah is a fifth-year PhD student in Valerie Eviner's lab at UC Davis. Her research focuses on multiple aspects of native versus exotic grass interactions, from competitive suppression through phenological overlap to spatial patterns to plant soil feedback. She is also on the board of CNGA. Thank you, Sarah. And in the pre-COVID world, as she says, she has enjoyed being a mentor with the restoration programs called SLU, that is the Student Landowner Education and Watershed Stewardship. Thank you, Sarah.
Hi, everybody. Today I'll be discussing a key aspect of my research that focuses on how temporal priority leads to native grass resistance to noxious weeds. And I know this is a grassland section, but as a brief intro, California grasslands are majority dominated by annual exotic species. However, they remain important ecosystems as they are habitat to a majority of the state's threatened and endangered species, serve as a global diversity hotspot, and provide us many ecosystem services we depend upon, such as livestock forage and water storage. And my research focuses on the interactions between our native grasses, which exist only in remnant populations and can be difficult to establish, and two groups of the exotic annual grasses, which I group based on phenology and flowering time, and are difficult to control and eradicate. The first group, which I classify as naturalized exotics and include avenas, bromes, etc., have been dominating for the last several hundred years and have an earlier phenology and flowering time. The second group is comprised of the noxious weeds, medusa head, and goatgrass that are currently invading, and they differ from the naturalized grasses in that they flower later in the season after the naturalized grasses have reached peak biomass and bloom. And medusa head and goatgrass are particularly nasty grasses that form non-palatable monocultures, causing great economic and ecological damage. And my research today elaborates on some knowledge that forms the basis of native grassland restoration. Because the exotic annual grasses germinate earlier and are faster growing, they often outcompete our slow growing natives. And this is why restoration practitioners focus on extensive weed control before seeding native grasses at a site. And this concept in ecology is called priority effects and can be applied to more than just grasslands. Priority effects are essentially when who arrives first matters. So the first to arrive has first access to those resources, allowing the species to grow and establish before other competitors arrive, resulting in a reduced population size of the new arrivals. So who arrives first determines the future community. And giving priority to natives often leads to success in the short term in restoration, but it's still kind of unknown how long these effects will be lasting. So my research explores if we can predict how these three grass groups will interact over a long period of time, because long-term interactions will determine the success of native restoration and noxious weed control. And unfortunately, making predictions about the future composition is in general difficult in our annual grassland system due to the high variability we see from year to year. And this variability is complex. On an individual species level, seed production and seedling survival, which are integral to an annual grasslands composition, depend on factors such as litter buildup, competition, and composition. And so it's just difficult to predict in what proportion species will be present in the following year based off of what we currently have. And as annual precipitation is such an important driver, we're expecting even more certainty in how our grassland composition will change as our climate is starting to shift towards extremes. In the last 10 years, we had a historic drought that was shortly followed by a historic wet year. So being able to make long-term predictions is even more important as we expect more extreme climatic events to occur. But maybe focusing on this group level might be able to help us make these long-term predictions, especially when it comes to sucking exotic grasses out by phenology. So Medusa head and goatgrass tend to bolt and grow to full size after the rains have ended, when soil water becomes limited and the naturalized exotics have finished growing. But adult native perennial grasses have already established deep roots that are actively using the soil water at that same time. And so due to this similar phenology, we can predict that if the natives are given priority, they might be able to successfully outcompete Medusa head and goatgrass for that soil water. So to boil my conjecture down to a concrete question, I'm using a 12-year data set to ask, how long do priority effects last for native grasses? And there's two aspects to this question. The first is, how long do the natives persist or last when being seeded first, but then left alone? And to apply this to the restoration, we ask, after maintenance site, how long will the natives survive or stay at a successful cover percentage? And the second part of this question is, how long will the natives resist invasion? So after maintenance or weeding ends at a site, how long until either the naturalized exotic or the noxious wheatgrasses invade? And is there going to be a difference between those two? And the final question I'm going to explore is, what role does extreme drought play? Does this extreme stressor disrupt any of these priority effects? And as more droughts are expected in the future, we need to be able to better predict what the long-term consequences of drought are. So I tested these questions using a larger grassland experiment set up in Davis, California in 2007. Bare ground was prepped, and in 1.5 by 1.5 meter square plots, each of the 3 grass groups were seeded alone. But for today's talk, I'm focusing on plots in which natives were planted alone in a mix, meaning that they were the first to arrive and thus given 1 year of priority over that space and resources. Other plots were seeded with the different combinations of the natives and the 2 exotic groups. And so they arrived at the same time, and natives were not given priority. And as naturalized exotics and noxious weeds were seeded elsewhere at the site as part of a larger experiment, they were allowed to move throughout the site and invade plots that they were not seeded in, such as the native mix priority plots. And then community composition was measured annually for 12 years. So going back to my first question, how long will natives persist when planted first? There's 2 results that we can look at. Does being planted with priority lead to the highest cover compared to being planted with the exotic groups? And then also, do the natives persist long-term when they might be pushed out or kept low when planted with those other exotic groups? And so looking at this graph, the green line on top represents the plots in which the native mix was seeded alone with priority. And the other colors are the treatments when the natives were seeded at the same time as the exotics. And the y-axis is the total native percent cover in each of the treatments. And we find that pre-drought, being seeded with priority led to the highest cover. But then the drought came and cover decreased in those native plots. But post-drought, native cover became more variable, and being seeded with priority, again that green line, only led to higher cover some years, and only ever when compared to the 2 treatments that contained the noxious weeds. Lines are significantly lower in cover, in native cover, than the blue and green lines. And it's really interesting that native cover in all of these treatments is rather high in the long term, at least 25%, and sometimes going up to 100%, meaning that native perennials do persist in the long term, even through extreme drought. So moving on to my second question, how long do natives seeded with priority resist invasion? And we're going to look at whether the invading grass groups are consistently low in these resisting native planted plots throughout the duration of the experiment. And we find that pre-drought, in the beginning of the experiment, native seeded priority plots were resistant to both naturalized exotic and noxious weeds. As you can see, these really low lines right here. These are the blue line represents the cover of the naturalized exotics in these native priority plots, and the orange line represents the cover of the noxious weeds in these native priority plots. And during drought, the natives lost resistance to the naturalized exotics. But importantly, and which is really cool, the native priority plots remained resistant throughout the drought and afterwards to the noxious weed invasion, keeping them at very low levels if present at all. And so remember, the noxious weeds used soil water later on in the season when it's now a limited resource. So these results follow our prediction that the natives can outcompete them due to having similar phenologies. And this last graph is sort of an aside that I thought would be useful information to share that highlights the importance of diversity in our planting mix. This graph shows the individual species included in the native mix. And it's each of these lines is each individual species and their cover in these priority plots. And the very first year, cover was completely dominated by our annual forbs, but then pre-drought, native cover is pretty much just Elmus glaucus. You can see this green line right here. But then with the drought, Elmus glaucus really did crash a little bit. And post-drought, Stipopulchra and Elmus triticioides to an extent increased in cover to come to a co-dominance with Elmus glaucus. And so this is kind of just more evidence that diversity in our planting mix is necessary to add resilience to extreme stressors such as drought. So to summarize my findings, the priority effects of seeding natives first lead to short-term success. Natives have the highest cover when planted alone and are resistant to invasion of both exotic grass groups. However, due to the disruption of drought, priority effects have mixed long-term success. For native cover, priority effects sort of weaken or disappear, meaning that being seeded alone doesn't lead to such a drastically high cover anymore. But still, being seeded with priority over those noxious weeds and Medusa head and goat grass is still important for highest cover. And the natives lose their initial resistance to the naturalized exotics, which is not much of a success, but it is a success that they maintain resistance to our noxious weeds. And to end, what are the management implications of these findings? You know, granted, my plots are only 1.5 by 1.5 meters squared, but I hope that they can, inform restoration management in some way. And so grouping our exotic annual grasses in terms of phenology is important because natives remain resistant to their phenologically similar noxious weeds with whom they can compete for the limited water. And this suggests that native restoration can be a key action to prevent invasion in vulnerable areas. And also because native cover is lowest when noxious weeds are present in the very beginning and when they were seeded with each other, we may need to prioritize control of the noxious weeds before we seed the natives. And then lastly, my results underline the resilience of native grasses. Once we get them in the ground, they weathered extreme drought events and can co-dominate with the normally dominant naturalized exotic grasses. And the diversity of the planting mix is important and necessary for this resilience. And so this just supports in general the need for more extensive native restoration throughout the state. And with that, I'll end by acknowledging the Vernal Lab and all of the funding sources that made this large-scale project possible. And now I'll be open for questions.
All right, thank you, Sarah. So attendees, if you'd like to ask questions, now is the time. This is great info. You know, as land managers, this is info that we can, you know, definitely use for planning purposes, you know, as far as what we're going to do prior to seeding or, you know, maintenance-wise. But I had a question for you. And I know there's so much you can do within a research project, but have you looked at a difference effect or success, I should say, based on the seeding rate? So for example, if you seed, you know, the natives at, let's say, 10 pounds per acre versus 60 pounds per acre, you get completely different results.
I think we didn't test seeding rates in this experiment, but I would guess that seeding natives more densely would probably be more helpful just because they are such slower to germinate.
Let's see, question from Elisa. Oops. What quantitative sampling method did you use to measure cover, and what were the pros and the cons?
So can you repeat the second part of that question?
Yes. So basically, how did you sample? What method did you use to sample, and to measure the cover? And then what were the pros and the cons of that method?
Oh, okay. Yeah. So we use we do visual estimation to measure percent cover, and we use the Daubenmeier bin method. So basically, it's less than 1%, 0 to 5, 5 to 15, 15 to 25, and so forth. And so that a benefit, so basically we say, oh, the cover of the species falls into this. And then when we do our analyses, we work with the center point of that bin. So if it was 15 to 25, it'd be like 20 something. And a pro of that is that it allows multiple people to be able to measure percent cover, because there was a study done that more people can agree on a bin rather than an exact percentage. But as that, we don't get as precise of data.
Yeah. Thank you. No other questions so far. I just want to make a comment, and this is actually a great study. And, you know, I think that you're able to do this for 12 years is pretty amazing. And that really gives a, you know, long-term idea of what's going on. And I know it was tough years because we did have that drought. You know, right about the same time, I was personally doing some studies on forb. California native flowers, because on my reserve, we were going to plant over 100 acres on them. And so we had to do a few trials on the density, species, and so forth, and kind of about the same thing, you know, for you, that's when the drought happened. So we had to redo that a few times. But like what I've noticed, at least on my grassland, is, you know, I mean, we do as much weed control as we can beforehand. And no matter what, the noxious weeds come anyways. But long term, especially with the Stipa pulchra, for example, the purple needlegrass, I see the weeds kind of going away on their own. So I don't know if it's specific to the Stipa, but you can, you know, the Stipa really starts dominating, and the non-native annuals start dissipating a little. I don't know if you've noticed that also.
So I think one thing is that we do notice a big difference between, you know, the Venus and Bromes and then that group of goatgrass and Medusa head. So the goatgrass and Medusa head did crash in general during the drought. And then they kind of bounced around after the drought. And so, but then also we had part of our larger experiment is that we have drought enclosures. So we're doing like these same species plantings, but subjecting them to a drought treatment. And the Medusa head and goatgrass basically all but disappeared completely wherever they were seeded in those drought ones. Whereas the naturalized exotics like Avena and Bromus, Diane just loved the drought and they kind of like really rebounded. So that's kind of a difference that we did see. And then, but the natives kind of just were steadily increasing throughout that time.
In fact, that's more or less similar question, but did you find the drought was an unexpected benefit to the study, getting to see its effect on density? So the drought kind of helpful, you think, to show the higher density of natives?
Is it helpful?
Any benefits of the drought?
I think, I mean, this is just all speculation on my part, just like looking at this data a lot, but it did seem like because the drought, like a little bit of drought does seem to be good for the natives at our site, because and then it was shortly followed by really wet years, so the natives just kind of like rebounded so well. And they rebounded, it seems like faster than the noxious weeds were able to rebound. But you know, if the drought was extended by even more years, then maybe the natives would have gotten too low of a point where they might not have rebounded, because they were getting really low with that drought.
Right.
Yeah.
All right, that's about our time. There's thank you, Sarah, very much. There's a few more questions in the Q&A, so if you don't mind answering those, that'd be fantastic.
Yeah.
All right, well, thank you so much.
Bye.
All right. Our next presenter is Christopher Hart with BLM. He's going to talk about goat grazing on coastal grasslands. So Christopher has he's a biologist for the Bureau of Land Management, and he works at Fort Ord National Monument. He's been working there since 2017. He finished his Master's of Natural Resource Management for Oregon State University in 2020. And he has been working with the BLM to manage their goat grazing program and coordinate volunteers to restore coastal grasslands on the monument. Chris?
Cool, thank you, JP. You can hear me okay?
Yes.
Cool. So as JP mentioned, my name's Chris, and today I'm going to talk to you about the way that we've been managing our coastal grasslands for the last 20 years and the different partnerships that have helped us along the way. The objective of the study is to simply summarize the 20 years of grazing history on Fort Ord National Monument, which has largely been between a sheep grazing study from 1998 to 2015 and several different types of goat grazing studies from 2015 through 2020. A little bit of background on Fort Ord National Monument. Fort Ord National Monument is located in Monterey Bay, California, and it's approximately 15,000 acre of public land. On Fort Ord, we manage maritime chaparral habitat, oak woodland, and 2,500 acres of coastal grassland. And then you see our study plot locations. We're located in between three major urban communities of Monterey, Marina, and Salinas, and we essentially manage the monument for conservation and recreation activities. Before being managed by the BLM, it was an active military base from shortly before World War I all the way to the end of the Korea War, and we have a long history of military activity on the monument. But now we manage it for conservation and recreation activities, and we have over 85 miles of road and trail, and a lot of those being in the coastal grassland areas where we have the sheep and goats graze. And so when we manage our coastal grassland habitat, we have two essential management goals. In the grassland themselves, we want to reduce the residual dry matter to being to 1,200 pounds per acre based off of that Bartholomew study. And that's to reduce the risk of fire to the surrounding urban community, but also open up the habitat so we increase the abundance of our native bunchgrass population and the native forb species. And the area of the coastal grasslands that have been intruded on by coyote brush scrub, we want to reduce that encroachment of coyote brush scrub and increase the abundance of native species in those areas. And the reason why this is so important, and the reason why we care so much about managing for the bunchgrass population, is that these bunch grasses are perennial grasses that can live up to 100 to 200 years old, and they grow naturally spaced apart in the grasslands. And there's a lot of open area in between them, which has a natural risk of fire to the surrounding community, and also leaves a lot of room for wildflowers to grow, like these non-native ones and potentially native ones. When we get an influx of annual grass and somewhat even coyote brush, they start to outcompete these native forb species for both competition for sunlight and for nutrients. And eventually when you get peak fire season, such as in September, it turns into this giant thick thatch layer, which is a huge fire risk to the surrounding urban community, either spreading a fire to the homes or having a fire get caught on from a fire from the homes. The same thing is true for the coyote brush. Even though it's a native habitat in California, it's kind of ubiquitous to the state and isn't quite as native or as diverse as our coastal grasslands. And what happens is that the coyote brush comes from these pockets of coyote brush scrub where it naturally grows and spreads into the coastal grasslands when you don't have enough disturbance. And eventually it completely transforms the habitat where you no longer have a grassland ecosystem. You just get this brushy mat of coyote brush, which is a huge fire risk because it's brush, but you also lose out on a lot of that native plant diversity. And so what we've been doing since 1998 is trying to manage the grasslands to get rid of the annual grass and get rid of the coyote brush. And initially we started it with a sheep partnership from 1998 to 2015, where we use sheep to manage the grasslands. And then in 2015, at Central Coast Invasive Weed Symposium, we learned that goats could actually be used to manage our brush encroachment in the grasslands, and they were willing to use fencing. So since 2015, we've been using the Goats R Us company as a partnership to graze our grasslands. And essentially what we've been doing is using these animals to graze the grasslands in 3 different ways, where we have sheep just roaming the landscape from 1998 to 2015 without any sort of fencing. And then with the Goats R Us company, we've been able to partner and manage them in a way where they're doing a similar grazing style to the sheep, but also can be targeted into very high specific priority areas in 1/2 acre to 2 acre pens until they eat all the vegetation and the stubble height is 3 to 4 inches. And it's because the goat operation provides us the ability to target goat graze that we've been using them since 2015. And the way that we've been monitoring our progress with the sheep and with the goat grazing study is through 2 different types of projects or research projects that I'm going to share the results for you. And so the first set of research project is 9 paired of monitoring plots measuring fuel acclimations in the grassland, or the fuel hazard or the fire hazard to the urban communities down over here, and a plant diversity project in the coyote brush scrub where we're measuring the goat's ability to remove the coyote brush scrub and if there's any diversity changes. And the first thing I'm going to talk to you about with this presentation is the results of that coyote brush scrub study and how we're monitoring for brush reduction and for plant diversity. And so in an area called Pillarcitos Canyon, we have 6 paired of monitoring plots. We're measuring the percent foliar cover of coyote brush, annual grass, other non-natives, bunch grass, and other native herbaceous species. We actually started the study with over 20 categories, a lot of them being noxious weeds, but we didn't have any noxious weed show up in the control or with the graze. So we learned very quickly off the get-go that we didn't have any noxious weeds such as harding grass, yellow star thistle, Italian thistle, or anything nasty get introduced by the goat grazing. And we've been monitoring these research plots by both the grazed plot and ungrazed plot throughout the study. And the whole reason why this project is possible is because of our partnerships that we have with the local university called Cal State University Monterey Bay and the Goats R Us company who provides its goats to graze these landscape. But if it wasn't for the university students, it would be a pain for us and really no way for us to conduct these research projects because we have 6 pairs of monitoring plots, and each of them has 3 transects that we measure percent foliar cover along these 10 meters transects. And it's with brave volunteers like Maddie Slavin over here and an unknown volunteer in the background that we're able to do these results and collect all this data. And it's a lot of fun for us, but we're hugely indebted to our partnership with the university. Otherwise, we wouldn't be able to conduct this research experiment. For analyzing this data, I'm displaying the raw data, such as the mean and median, graphically using box plots. And the actual statistical analysis itself was a non-parametric stat test that I can explain in a question. But it essentially involves me simulating the data and resampling the data 10,000 times with replacement and comparing the significant difference based off of non-overlapping confidence intervals. And so with that out of the way, I want to talk to you all about the results of our brush grazing study and everything that we learned so far. And so all of my results are going to be displayed to you box plots given in this PowerPoint. I'm going to briefly explain what all is on this plot. On the y-axis, you have percent cover ranging from 0 to 100%. On the x-axis, you have treatment, graze control, so on and so forth. on top of the y-axis, you have the timing that we sampled. So we started in winter 2014, and then we began to sample spring 2013, and every spring there on out. Significant difference is denoted by this teal highlight, and the sample size is also up top, and the mean is represented by this bold red line, and the median is represented by that bold black line, and the plant individual that we're in question with is at the very, very top. And so the big takeaway here is that this is a boppy percent cover, percent foliar cover, that's a coyote brush. And what we noticed is in between spring 2014 to spring 2015, the goats completely devour all the coyote brush, and we completely lower the percent foliar cover of coyote brush scrub starting in 2015 and keeping that trend to 2016, 2018, 2019, and 2020. And so what we learned through this monitoring study is that the goats are able to reduce the percent foliar cover of coyote brush scrub, which is really exciting for us because it's one of our objectives. The inverse is true for the annual grass. As soon as you remove that coyote brush scrub, the annual grass starts to increase like crazy. And so starting in 2016, we had huge increases of percent foliar cover of our annual grass species, such as Bromus deandrus or wild oat grass. When it comes to non-native wildflowers, such as like Vicias or Geranium mole, we don't see a very strong trend. In general, the graze plots are trending upward, where you have more abundance of, or percent foliar cover of the non-native species, but there isn't a concrete trend, and so we can't say that the goats are introducing more non-native species compared to the control plot, because it doesn't happen every year. Fortunately or unfortunately for us, there's almost no trend with the bunch grass, because with every year except for spring 2016, there was practically the same amount of bunch grass present in the graze plot compared to the control plot. And so the goats aren't really having an effect on the bunch grass distribution. And for our other non-native herbaceous species, we're still trying to scratch our head about what's happening here, because there's absolutely no trend. And it kind of looks like a roller coaster where sometimes we have more native species present and sometimes we have less native species present in the graze plots. But that overall allows us to conclude here that when we have goats grazing this coyote brush scrub in this area of formal coastal grassland, we're reducing the coyote brush significantly and we're increasing the annual grass significantly. We don't really have a trend of non-natives, even though it's trending upwards and we think we're getting more weeds in there, but we're not affecting the bunch grass at all, and we're not affecting the native forbs. And so essentially we know that we're converting coastal grassland, or we're converting coyote brush scrub into coastal grassland, which is achieving our management goal. And it's really exciting for us that we were able to remove coyote brush and get it to look more like a grassland ecosystem again. And so now that we understand how to monitor this coyote brush piece, the next step that we have to ask ourselves is, how do we monitor the grasslands once we remove the coyote brush piece from it? And we've been trying to answer this question through two ways: through a plant diversity study with the university, which we're still developing the results for, but also through a fuel acclimation study that's been happening since 1998 all the way through 2020. And so now I'm going to talk to you all about how the goats and the sheep are affecting fuel acclimations in coastal grasslands. And so essentially what we're doing is measuring the dead annual herbaceous presentation between the graze plots and the ungrazed plots. We had 9 pairs of monitoring plots. And again, this is a whole project that's driven by partnerships. We're partnered with the sheep company, the goat company when the goats were introduced. And if it wasn't for the brave university students, again, out there collecting data, like sometimes 2 to 3 times a year, we'd never be able to get this data around her, because at each one of these monitoring plots, we're collecting 6 samples of biomass randomly determined within the plot. And so each treatment gets 54 samples by the time we're up and running with the study. And if we didn't have students, like these students pictured in this photo, there'd be no way for us to collect this data. And we're hugely indebted to this university for being able to conduct this study. So now I'm going to share with you the results of this biomass study. These are, again, box plots. The only thing that's really different between the ones that you just saw is that now we have PG representing passive grazed and UG representing ungrazed or control. The y-axis is now scaled from 0 to 10,000 pounds per acre, and this y-axis is going to change in between the plots, so just keep an eye out for that. Significant difference is still determined by the teal highlight, and the sample size is still up top. And this is just all of the results from our passive sheep grazing study from 1998 all the way to the end of 2015. And what we noticed from looking at these box plots is that there's practically no difference between the passive sheep grazing and the ungrazing. In general, on most years, it's slightly lower with grazing compared to not doing anything at all, but we're not seeing any significant difference when we've grazed sheep from 1998 through 2015. And so the sheep weren't really having any significant difference on the grasslands for reducing field acclimations and reducing that fire risk. When we look at passive goat grazing from 2016 to 2019, we kind of see a similar trend. Some years it's a little bit more consistent. There's a significant difference, but they're basically having the same exact effect as the passive sheep grazing. It's lower compared to the control, but it's not always lower, and it's not meeting our management goal of 1,200 pounds per acre. Where we see really exciting results, and this is my favorite side of this whole thing, is when we start using targeted goat grazing to manage our field acclimations. When we force the goats into a very small pen and force them to eat all of the vegetation until there's only 3 to 4 inches of vegetation remaining, we get closest to meeting our management goal, and we have the most reduction with this AG category, which is our targeted goat grazing. And as soon as we target goat graze at the start of the year, we see that those reductions take place through the growing season until the goats unfortunately leave in June, and then we no longer have any reduction. But as soon as the goats come back, we see that we start having more reduction, and when they target graze a subset of the plots in December, we get back to getting really, really close to our management goal. And so with our field acclimation study, what we learned is that between all 3 styles of grazing, we're not meeting the 1,200 pounds per acre management goal. But passive sheep grazing is better than doing nothing, which is really good to know. So we are, like sheep, we're reducing field acclimations, just not to our goal. Passive goat grazing is having the same effect, but the advantage of the goat grazing is that they can be targeted goat grazing and become the most effective grazing treatment. And even though we're not meeting our 1,200 pounds per acre, we're getting close. We're getting an average of 2,000 to 2,500 pounds per acre, which may be more fitting for our coastal grasslands because every grassland system is unique. And so the overall conclusion that we're learning from this is that targeted goat grazing is the most desirable management practice, and maybe the 1,200 pounds per acre management goal is too low, and we need to increase it based on the ecosystem and forward. The thing I want to point out here is that if you extend the length of time the goats are on the monument, you actually don't reduce field acclimations any further because the goats stop eating the grass when they're seed heads, and we didn't believe it until we witnessed it for 2 years in a row. And so we can't just leave the goats on all the way through June and expect our field acclimation study to be successful. As far as the coyote brush scrub studies is concerned, we know the goats are reducing coyote brush encroachment into coastal grasslands, but the growth of annual grass and other non-native vegetation is warranting new management practices. And so we need to start thinking about how we convert these coastal grasslands into native forb habitat, into more native bunchgrass habitat. And so the next steps for these projects are to simply continue to goat graze the coyote brush scrub, continue to remove it down and convert it to coastal grassland, but also include additional monitoring plots that aren't pure coyote brush scrub and start conducting more plant diversity studies, which we've just recently started doing, but kind of expanding that out so we learn more about how the goat grazing is affecting the plant diversity in the grasslands. As we do that, we need to continue to target goat graze priority areas before the goats leave in April, but also consider using alternative management strategies to further reduce the field acclimations in the grassland, to open up the habitat so native forbs can grow, and to reduce the fire risk. And some ideas that we're debating and trying to test the effect on are mastication with heavy equipment such as milling, or using targeted cattle grazing like they do at the Santa Lucia Conservancy, or maybe introducing some other animal depending on what our stakeholders and other people have opinions on. And so that's the presentation that I had. I just want to hugely acknowledge the Goats Are Us company and all the goat herders and the sheep operators for everything that they've done, and all of our university partnerships who've made it possible. The BLM is not a research institution or agency by any stretch of the imagination. And so it's because we have a partnership with the university that we're able to conduct these research projects on our land. And then if there's any questions, I guess you can ask them in the Q&A, or you can send me an email at char@blm.gov. And thank you, everyone.
All right, thank you, Chris. Questions? There's only one question for now. So let me ask you something very quick. One, just based on actuality or current news, has the site been affected by the fires down there, or you guys were far away?
No, it got really close, where like the river fire got within a couple miles of Fort Ord, but it never jumped the highway and started Fort Ord on fire. So we were closed for a while because of the fire risk, but it never actually entered our property. And so we didn't have any effect by the fire.
Okay. And then is fire a tool that you have in your toolbox to manage the land?
Right now it's not, because the urban communities are very afraid of fire, and they fight us when we try to do a prescribed burn, and we don't have the budget to conduct prescribed burns. It's extremely expensive. And so right now it's not, but if things change in the future, like maybe if the political climate changes and people are more open to prescribed burns, it might be something we're able to do.
Okay. And then question about sheep grazing. So sounds like as long as you have, goats, as long as you have goats to browse on the coyote brush, things are great. So hopefully you have goats for a long time, but what happens if all of a sudden, for whatever reasons, you cannot use goats anymore? Would the coyote brush come back and would I set you back to kind of how it was before?
Yeah, so we think if the goat partnership ended for who knows what reason, then the coyote brush would slowly start growing again and it would slowly start intruding in the landscape. The goats don't kill the coyote brush, they just kind of stunt its growth and like eat it all the way down to the ground. But I mean, the coyote brush is so good at surviving, it grows back after you mow it down. And so after several years, we would see more coyote brush, at which case we'd probably have to start masticating it with heavy equipment and use some other alternative management strategy to get rid of it.
Okay, thank you. Since you're talking about this, question from Bruce Delgado. Can you discuss mowing versus grazing, sorry, plus mowing as an alternative?
And so the big difference between mowing versus grazing is that when you mow, you're kind of obliterating everything in the landscape and you're mowing down the grass and you're mowing down the coyote brush, and it's much more consistent, but it's also a little bit more destructive because you're hitting everything. Whereas when you have goat grazing, it's more of a natural disturbance that's somewhat adapted. Oh no, my internet. It's somewhat adapted to the environment. And the goats really only target like the grasses and the vegetation and the coyote brush. They don't have as consistent trend. And so it creates more of a mosaic when you goat graze compared to just like this homogeneous removal of coyote brush. And we think this like mosaic of sometimes you have big coyote brush and sometimes you have small coyote brush after the goats creates it more beneficial for the environment and for like the different users of the habitat compared to when you just mow it down. So we've been hesitant to mow it because we think it would change the functionality of the ecosystem.
Great answer. Thank you. How about cost? How does like grazing cost compare to other management techniques?
So right now, the Goats R Us company, we have a partnership with them where they graze on the off season from November to April for free. And then come starting at fire season, when they get paying clients, they leave like forward, like at the end of April. And so right now it doesn't cost us anything to have goat grazing. If we were to continue the service through like May and June, it would cost us $1,000 an acre to graze because the Goats R Us company charges that for paying clients. And so right now it's practically nothing. We've been debating about trying to find like other grazing operators who'd be willing to do a same or similar agreement during the summertime, like when all the grass is done and there isn't very much feed for their animals, if they'd be willing to do something that's low cost. But right now there's no cost associated with the Goats R Us company.
Okay, that's great. So often you have to pay them to graze. That's fantastic. Let's see. Okay, that's from Joanna. Based on one of your previous slides, just a clarification. In passive grazing, the goats successfully decreased the coyote brush, but they didn't like to eat the annual grasses and form.
So what we think, we've never passive grazed the coyote brush, except for like just looking at them. The goats won't actually like target down the coyote brush unless we lock them in like a 1 acre pen and force them to eat all the coyote brush, because the goats are kind of picky on what they eat. And so some days they really like the coyote brush and other days we kind of have to leave them in there until they eat all the things they like, and then they'll eat the coyote brush. And they do eat the annual grass and they do eat the bunch grass and other things. Just when we did our study, we weren't finding like a month after the growing season has started that we saw any significant difference within those other categories, except for coyote brush and except with annual grass.
Excellent. Just one question related to one of the graph slide. Did you say you took like 10,000 samples?
So it was a bootstrap simulation analysis. So each plot had 3 raw samples and we had 18 samples total. And the way we got to the raw data was that we had 40 raw hits of data that got summarized into one of those like data points that you saw in the box plots. When I did the bootstrap simulation analysis, it's a simulation study. So I combined all of that data and then resampled it with replacement 10,000 times using statistical programming. And so I used a simulation analysis to get to that 10,000, but we didn't physically measure anything 10,000 times.
Okay, that makes sense. Question from me. So you guys have like sheep and goats coming in. Do they clean them before they get onto your site?
Yeah, so we have 2 methods of it. We physically visit the goats wherever they're going to be before they come here to Fort Ord. And then we look at what they're eating to see if there's any noxious weeds like yellow star thistle, Italian thistle, anything else we don't desire. And then usually almost every time we ask the goats to feed them alfalfa for 4 to 5 days straight to like clean out their guts a little bit. And then they get dropped off on a location on forward that's very easy for us to find weeds if they were to bring them onto forward. So then we kind of follow the goats around and keep track of where the goat activity was and check it out with our invasive weed crew for like the next growing season. And so we try to take every precaution so we don't bring like yellow star thistle or purple star thistle onto forward.
That's fantastic. Yeah, this often we forget, but it's actually very important. Now I have one section of my reserve where, because I use cattle grazing quite a bit, and at one time, which was the first time, didn't even think about it, and I started having weed infestation that I didn't want to.
Oh no.
So, anyways, all right, that concludes the time. Chris, thank you so much.
Yeah, thank you.
If you don't mind going to the Q&A and answer what has not been answered, that would be fantastic. All right. Our next presenter is Julia Michaels. She's going to talk about positive impact of grazing vernal pools. And I gotta find you right here. So Julia completed her PhD in ecology at UC Davis, where she focused on research on strategies for restoring native vernal pool wetlands. Julia also holds a Bachelor of Science from, I actually don't know that one, sorry if I misspelled it, Wesleyan University, and currently teaches in the Environmental Studies Department at Sac State University. In her spare time, Julia leads trips with National Geographic student expeditions and works at the Miriday mobile plant nursery, connecting with the public about the fascinating world of native plants. Julia?
Yeah, so just to, can you hear me?
Yes.
Ah, okay. It says that my internet connection is unstable. So JP, please do jump in. All right. Well, thank you. My name again is Julia Michaels, and I'm very excited to get to present some of my research to you today. I, like Sarah and Brianne, also worked in Dr. Valerie Evner's lab and just finished up a postdoctoral scholar position in Emilio Laca's lab. Both labs focus on addressing key conceptual questions in ecology with clear applications for ecological restoration. And I grew up in California, but I've worked and studied restoration projects in other places like Alaska and Louisiana. And so as concerns grow about biodiversity decline, I've started to devote my research to understanding the patterns of biodiversity and the underlying processes that shape it. So I focus on these. So in this talk, I'm going to focus on the question of how we understand, restore, and manage biodiversity in ecosystems that vary spatially, so over space, and temporally over time. The research that I'll be focusing on today took place in an ecosystem that is both highly variable, remarkably diverse, and of critical conservation concern, which is vernal pools. So I'm sure most of you guys are familiar with vernal pools, and but just to recap, as you guys are well aware, California grasslands in particular are highly invaded by exotic grasses. And a large portion of the remaining native plant species exist in many small patches across the grassland landscape. So these patches occur because of some environmental factor that filters out invasive grasses and provides a refuge for native plant species. So vernal pools are examples of these refuges. Vernal pools are depressional wetlands with a hard soil layer underneath that causes seasonal ponding. And in the summer and fall, they're bone dry like the rest of the grasslands. But in the winter, they fill with water. And in some cases, these pools have been forming in the same spot for millions of years. And because it's too wet for upland and too dry for wetland species, this ponding filters out many exotic grasses, allowing native species that have evolved. And the plants that bloom in the spring, as you can see, form remarkable ring patterns as the pools dry down, which I'll get to more in just a second. So vernal pools are just one of those many ecosystems that we ecologists call patchy. So these patches can vary a lot in which species they contain. Each individual patch may not actually contain very many species, but the high variation between the patches adds up to a lot of diversity at the landscape level. And when I say a lot, vernal pools host over 200 species of native and endemic plants, which is disproportionately large considering how small of an area that these wetlands take up. The plants that live within vernal pools are highly sensitive to small differences in the timing of inundation. So this can be observed visually as the pools dry down in stages and different habitat zones emerge that host only certain species that are adapted to live in that specific set of conditions. So these habitat zones can be divided into pool, transition, and upland. So I have both a photo and a graphical representation. And so the pool zones hold water for the longest amount of time, and they host many vernal pool species. The upland zones are the outer edge of the pool that don't get inundated, and the transition zones flood and dry often during the season and host both upland and vernal pool species depending on seasonal precipitation. So there's high variation within vernal pools between the pool, transition, and upland zones. And then vernal pools also vary in size, shape, soil type, chemistry, and groundwater availability, which leads to high variation between the pools as well. So for example, these 3 vernal pools are adjacent to one another at my field site, taken on the same day, and clearly host very different floral patterns. So you can imagine these 3 graphical representations of these 3 photos. So despite their habitat value, vernal pools have been being lost at an alarming rate due to urban and agricultural development. However, there's still many left to protect and many that have been restored or created. The remaining vernal pools are almost all grazed, either for ranching or for management of exotic species in the uplands. And a previous study by Dr. Jamie Marty, who also came out of the Davis Ecology Program, found that livestock grazing can help maintain plant diversity in vernal pools, although the exact mechanisms have not yet been identified. This study, along with other work, has led to widespread shifts in how people think about grazing as a conservation tool in vernal pools. However, no studies have looked at the effects of grazing on this variation in plant communities, both within and between pools. So we're left with an important management question. How does grazing affect this plant diversity both within and between vernal pools? This is important for vernal pool conservation because land managers need to know how grazing affects the distribution of key species that they're targeting. For example, we'll see later on in this talk that the transition zones play an important role in hosting biodiversity. So a land manager might want to know, are they losing the transition zones within pools by these habitat zones? Or maybe seed dispersal, like we talked about in the last lecture, maybe that or preferential selection by cattle could be making the pools more similar to one another. So in addition to addressing these questions that may help us manage vernal pools better, they're also a great study system to test out important questions in ecological theory. So for example, one pattern that we see often in ecology is that the effects of environmental disturbances such as grazing, floods, fires, etc., can change with spatial scale. So specifically, we often find that disturbances can cause increases in biodiversity within small local patches, but not at the larger scales. So this has been seen across many ecosystems, including rainforests, temperate forests, and grasslands, both Music and Xeric. So several studies have suggested that this scale difference has to do with how biodiversity is measured. So vegetation lesson. So let's imagine that the orange circles are conceptual habitat patches that could be located in any ecosystem. So the smaller circles within the patches represent individual plants, and the colors represent different species. You with me? All right, let's go. So we call the diversity within each patch alpha diversity. So this patch has an alpha of 1 because it has 1 species. This patch has an alpha of 2 because it has 2 different species. And this has an alpha of 3 because it has 3 different species. So that's alpha diversity. And then the diversity of all the patches together across the landscape is called gamma diversity. In this case, the gamma diversity is 3 because there are 3 species total. The variation between the habitat patches is called beta diversity. So the beta diversity is the link between the alpha and the gamma scale, and is often calculated as the gamma divided by the average alpha. So we can say that altogether the beta diversity, or the value of the variation between the patches, is 1.5 in this conceptual system. So beta diversity is important because it can reveal the spatial scaling of diversity losses and gains. And it's a very important component of many different types of ecosystems. And it's important for management because, for example, if all 3 patches were the same, a land manager wouldn't have to be concerned about losing species if one patch was lost. But in a system with very high beta diversity, it may be critical to conserve all the patches to protect all the species. So I'm interested in how disturbance affects these 3 levels of diversity: alpha within, beta between, and gamma overall. So imagine we have 3 patches with 1 species each and a gamma of 2 species. So the blue arrow could be any type of disturbance. So remember, this could be fire, grazing, or flooding. In this case, of course, I'm studying the disturbance of grazing. So I looked into the literature and found many different possible responses of alpha, beta, and gamma diversity to disturbance. In each of the 3 possible scenarios that I found, disturbance disperses species between patches, aka the beta goes down with disturbance. So you can see here that the patches in this conceptual scenario are now identical. Depending on whether the land manager wanted to increase or decrease the variation between the habitat patches, this lower beta diversity could be good or bad. Grazing can also add species to patches by bringing new plant species from outside the system. If grazing also adds 1 new plant species to the landscape, then the alpha within the patches goes up, gamma across the landscape goes up, and the beta diversity goes down since the patches are now more similar. Or rather than bringing new species, disturbance could disperse species between patches with no effect on the total number of species at the gamma level. This could be tricky because if the land manager only looked within patches, they might think the diversity at the gamma level had increased, when in fact it had really stayed the same. And again, beta again would go down. And then finally, grazing can actually remove species from the landscape if they're intolerant to grazing, while still dispersing species around between patches. Now alpha goes up within each patch while the gamma actually goes down. This scenario would be very tricky for a land manager because if they were to look within one patch, they might think diversity is going up, when in fact they're losing a species at the gamma level. So these are all possibilities of what could be happening in my system with vernal pools and grazing. So I identified 2 levels of alpha, beta, and gamma diversity in my vernal pools. So the first one is very similar to our conceptual orange circles. The diversity within a given vernal pool is alpha, the diversity at the whole pasture scale is gamma, and the variation between the pools is beta. The beta diversity is important, again, because a manager would need to know how unique each pool is across the landscape and whether grazing is maintaining or decreasing this uniqueness. It could be helpful, for example, if the manager had to decide whether some vernal pools could be given up for housing development without losing any species at the pasture scale. We can also look within the pools at each individual habitat zone. Alpha would then be the diversity within a given zone. Gamma in this case would be the whole pool, and then beta would be the variation between the zones of the pool. And this type of beta diversity is important to know if the unique habitat structure within the vernal pools is being maintained, or if the cattle might be breaking down the differences between the zones. So again, I wanted to know about diversity of these 3 levels, alpha, beta, and gamma. So first I wanted to know if grazing changes alpha diversity within pool habitat zones and within pools, whether grazing changes variation between the pool, transition and upland zones within the pools, and between pools as a whole. And finally, whether grazing changes diversity at the whole pasture level. So my study site was located in Sacramento County on a reserve for natural vernal pools, and a fence line around a large reservoir separated pools that had been continuously grazed from pools that have been removed from grazing for over 40 years. So you can see the pools from the satellite imagery here, and there's also pools inside the fence, although they're harder to see. So inside was excluded, outside had grazing. I chose 15 grazed and 15 ungrazed pools, and since we know there's so much variation between pools, it was important to carefully pair them based on size, shape, soil type, and topography, which other previous studies haven't been able to do. And I monitored in the spring, in the peak flowering season, for plant species richness, evenness, and native cover. And I structured my sampling to look at those pool, transition, and upland zones separately. And as you can imagine, this was an enormous field endeavor. And in order to achieve this, I recruited and assembled a team of individuals from very different backgrounds, including Sacramento City College, UC Davis undergrads, ranchers, and ecological consultants, some of whom had never gotten to do ecology before. So it was really fun. So the results of this research were very encouraging. I found that livestock grazing decreased the abundance of locally dominant species, mostly exotics, and increased the abundance of locally rare species, mostly vernal pool wildflowers. So this is important because we're most often concerned about losing locally rare species to more locally dominant ones. Okay, so what about the diversity in the alpha, beta, gamma stuff? So we can start by looking at the smallest alpha scale, or within the habitat zones in vernal pools. So I'm going to explain this graph. On the x-axis, there are 3 levels of diversity: diversity in the zone, diversity at the whole pool level, and diversity at the pasture level. And then on the y-axis, we have the values in terms of diversity. So the 2 lines are grazed on the top and ungrazed on the bottom. So we found that grazing led to significant increases in species richness, species evenness, and native cover at that local habitat zone scale. So you can see it's significant stars indicating significance at that local scale. So we got increases in species richness, evenness, and native cover. And we looked specifically at each of the 3 habitat zones, the pool, transition, and upland, to see which zone was driving these differences. And we found that it was the transition zone consistently responding across all 3 metrics of diversity. And based on ecological theory, this is not surprising because often in ecology, we find that transition habitats, also called ecotones, are the most influenced by disturbance. So in our case, the pool zones, or the bottoms of the pools, are so inundated that grazing doesn't have much of an influence, since the most dominant exotic species are already filtered out by the standing water. And in the uplands, the grasses dominate and exert the strongest influence. But in the transition zones, both upland and pool species can survive, so they can be strongly influenced by grazing.
Hey, Julia,
okay, next we're gonna look at the diversity within pools as a whole. So we found, am I cutting out?
No, I say you have about 3 minutes.
Oh, I know. Okay, I'm finishing in pools, and we found that there were increases in diversity at the pool level as well. And then finally, looking at diversity differences between the habitat zones, so the beta diversity differences, we found that grazing did not disrupt those differences between the 3 habitat zones. And in fact, the plant communities were distinct when you looked at species identity as well. And we did find that beta diversity between pools as a whole were affected by grazing and reduced. So grazing made the pools more similar to one another. And we found that both with the species richness and also looking at species identity, which was in our using an NDMS plot. And then finally, at the whole pasture level, when you look at gamma diversity or species richness at the whole pasture level, you can see that despite the fact that grazed pools had more species, that does not mean that there were more species in the grazed pasture overall. So again, we see a pattern where disturbance is increasing diversity at the local scale, but not increasing diversity at the landscape scale. And so what we can conclude is that what's going on is that grazing is increasing species richness within pools, making pools more similar to one another, which makes it appear that diversity is increasing without actually adding any new species to the landscape. So that was the third hypothesis that I had about what was happening with disturbance. So it both addresses this question of what grazing is doing to the vernal pools, and also why we see scale differences between small scale and large scale grazing effects. So finally, my recommendation for managers is that the transition zones host a lot of diversity and are the most responsive to grazing. That livestock grazing can have positive effects on diversity and native cover at the zone and pool level, but not necessarily at the pasture level. That livestock grazing preserves the unique spatial structure within vernal pools, including the transition zones, while making pools slightly more similar in species. It's important to measure the effects of grazing disturbance using these multiple scales and metrics in order to see the real effects of what's going on. And with that, I just want to thank my funders and my numerous field assistants. And I think I went a little over time, so the timer stopped working. So I was winging it.
You're okay. Thank you, Julia. So I have a question for you. Did you fence like the perimeter of the site and allow the cattle to go into the vernal pool, or did you actually force graze the vernal pool? Meaning you only build a fence around the vernal pool.
So I actually found a natural.
Go ahead.
So I found a fence line that separated the grazed and ungrazed vernal pools for over 40 years. So I didn't actually do any manipulation of the grazing in this experiment.
Okay. Hey, Julia, I hate to tell you this, but we're losing you a tiny bit. If you turn your video off, the sound should be better.
Mm-hmm.
Don't mute yourself. Just turn the video off.
Had it.
Okay, so there was a question.
Okay.
Based on what you say, I don't know if you know that one, but was the seasonality, species of grazer, or grazing intensity analyzed? And if so, was it different among treatments?
So we had a conservation stocking rate. So it was about 1 animal unit per 6 hectares, which is pretty standard, I believe, for that area.
Mm-hmm.
And the grazing happened from November through June.
Okay, was that exclusively with cattle, or did you try other things like sheep and goats? And
it was exclusively cattle.
Okay, thank you. Next question. Was it difficult to distinguish the zones in the vernal pools?
Zones, you could go by the vegetation alone, but of course we were trying to do it based on abiotic characteristics. So what we did was we measured the slopes. So the bottom of the pool had no slope, and then as it slowly increased, that's how we found the transition zones. And we also used watermarks, which allowed us to determine how much water had been present in each of the zones.
Excellent. Next question. Was the site grazed before the study, and was a baseline conducted? So in other words, was it that diversity was actually increasing in the grazed pools, or was the diversity decreasing in pools when grazing was stopped?
So that's a good question. This site had been grazed for, as far as we know, over 100 years. And then this fence line had been built 40 years ago. So by the time I got to these pools, there had been 40 years of ungrazed, and then 40 years, or over 100 years of continuous grazing. So it's hard to know whether the lack of grazing, the long-term exclusion, was leading to a decline, or whether the grazing was leading to an increase.
Okay, and that's about our time. Thank you, Julia. If you don't mind, go on to the Q&A. There's at least one more question. a couple of questions that needs to be answered.
Great.
And thank you very much.
Awesome.
All right. Our next and last presenter is Brianne Palmer. She's going to talk about a BioCrust study. Brianne is a fourth-year PhD student at San Diego State and UC Davis. Her primary research is focused on characterizing the composition and function of BioCrust after disturbance and advocating for more awareness of BioCrust in grasslands. When she is not thinking about BioCrust, she writes for a couple of science blogs, plays with her cat, and bakes a lot of bread. You're funny. Brianne, all yours.
Okay, speaking of my cat, she decided to jump up right this second. So hi everyone, I'm Brianne, and today I will be talking about my research on this really cool microbial community, biological soil crusts, and how they respond to grassland fire. So first, I briefly want to discuss the importance of fire for ecosystems. Prescribed fire is a common management strategy for both forests and grasslands. And there are a variety of reasons to perform a prescribed burn. They're usually done to improve the habitat for wildlife as well as reduce the fuel load. And in California, indigenous tribes burn the landscape and many still orchestrate prescribed burns today. Wildfires are also an important aspect of the ecosystem, and many species rely on wildfires to maintain the ecosystem structure. However, wildfires are increasing in frequency and severity globally. Currently, California is facing an intense wildfire season, and we need to be aware of how these wildfires will impact the environment. We also know that both types of fires change the ecological community in a variety of ways. So, for example, in the world of vegetation research, John Keeley did a study in the mixed coastal sage scrub in Southern California grassland. In this graph, native cover is represented by the gray bars and the black bars are non-native cover. So this study showed that when these ecosystems received more frequent wildfires, there is greater percent cover of non-native plant species compared to sites that had less frequent wildfires. Alternatively, prescribed burns can be used to change the plant community and actually reduce the cover of non-native species. For example, in this study, we see that the percent cover of medusa head is lower in sites that were burned compared to those that were not burned. And this just demonstrates the variability of fire effects on the vegetation community. We also know that fire affects the soil microbes. Both wildfires and prescribed fire can change the species present and their abundance in the soil. Additionally, microbial biomass, shown in this graph, decreases as fire severity increases. And there are often stark differences between soil communities burned in a low severity prescribed burn and a more high severity wildfire. But many of these studies on the vegetation community and the soil microbial community neglect a key ecosystem engineer living on the soil surface. Which leads me to my primary research question: how does fire alter the microbial community living on the soil surface, specifically biological soil crusts or biocrusts? So biocrusts are present in California grasslands. We just may not see them if you're not looking for them. But recently they have been gaining more attention. So biocrusts are comprised of cyanobacteria, lichens, mosses, and liverworts, and they all live together and they're bound on the soil surface. So they're essentially this microbial community that you can see without a microscope. So for example, in San Diego, it's common to see these dark cyanobacterial crusts with these mixed lichen crusts. In Temecula, this is the coastal sage scrub system. Again, we have these dark lichen crusts. Bakersfield, I found this dark moss crust. And then in Davis, this is actually one of JP's plots. There's this moss crust growing right next to a stipa. And then even in Napa, I found this lichen crust growing in the serpentine grasslands. So biocrusts are important to the ecosystem and may modulate plant growth. They can either inhibit or facilitate plant germination. They change the soil moisture content and they reduce erosion. They can also fix atmospheric nitrogen and carbon and are important in a variety of biogeochemical cycles. And most importantly for this study, they are directly exposed to fire on the soil surface. So when an ecosystem burns, they are on the front lines and likely more affected by fire than the deeper soil microbes. So in order to understand how biocrusts respond to fire, I work on San Clemente Island. So San Clemente is a naval base off the coast of San Diego and is the southernmost Channel Island. The plateaus on the island have a very diverse grassland, and the majority of the moisture is coastal fog. And these grasslands also harbor a diverse biocrust community, which no one's ever studied before. So this is a drier grassland, which creates these vast patches of soil crust in between the plants. So the soil crust can grow between the plants, adjacent to the plants, or even underneath them. San Clemente also has a recent and well-documented history of fires and pre-established plots, which allowed a good setting to understand biocrust response to fire. So on San Clemente, I have 3 sites. And just to orient you to this figure, the open squares are burned plots and the darker squares are controlled plots. So sites 1 and 2 were burned in prescribed fires in 2012 and 2017. Site 3A was burned in a wildfire in 2012 and then a prescribed fire in 2017. Then site 3B was only burned in a wildfire in 2012. And each prescribed burn only occurred in a 10-meter square area. The wildfire plots denoted here. Again, they were only burned in 2012, and then we selectively burned 10 more plots just north of them in 2017. And then on the other side of the road, the fire never burned, so those were the control plots. So each plot is 10 meters squared and divided into 4 quadrants, and within each quadrant, we placed a 1 meter square frame and measured the percent cover within that frame. So for all the sites, we measured percent cover of biocrust in 2018. So that's 1 year after the prescribed fire and 6 years after the wildfire. However, in 2017, we were only able to measure percent cover in sites 1, 2, and 3A because we lost the plot markers at the wildfire plots. They were likely washed out with the rain because that was a really crazy rain year. And there's also evidence of off-road vehicle use near the plots. Then in 2020, despite the pandemic, 2 of my collaborators were lucky enough to get out to the island and they were able to measure percent cover this past spring. Again, we didn't sample the wildfire plot. And then in 2018, I collected DNA samples from the plots that were burned and just the prescribed fire and just the wildfire. So this allows me to ask 2 primary questions. First, using the cover data, I can ask how does prescribed fire change biocrust cover over time? And second, using DNA, I can ask how wildfires and prescribed fires change the microbial community. So for my first question regarding biocrust cover, I predict that prescribed fire will reduce the overall biocrust cover because fire is a severe disturbance of the soil surface and will likely reduce biocrust abundance. Second, I predict that the control, prescribed, and wildfire microbial communities will be distinct. This is because fire, again, is a big disturbance of the soil surface. And even 1 year after the fire, I expect the communities to be different. And I expect to see legacy effects of the fire in the wildfire communities. So does prescribed fire change biocrust cover? It does, but the cover was different in the burnt plots compared to the controls, but not in the ways we expected. So here you can see site on the x-axis and total cover on the y. The red bars are burn plots and blue bars are the control plots. And we can see that across all sites in all years, there is greater biocrust cover in the burn plots than in the controls. So this was contrary to what we predicted, and there are a couple of reasons why we thought of to explain this result. So one possible reason is that there is a negative correlation between biocrust cover and plant cover within the burn plots, indicating that the plants and biocrust may be competing for space, light, or other resources, and the plants generally win. Therefore, after a fire, there are less plants and therefore more space for the biocrust to quickly colonize. So this is a visual example of what some of the quadrants looks like. So in the burn plots, we can see there's more empty space between the plants, and that empty space is eventually colonized by biocrust. In the control plots, the plants are more dense, there's a lot of thatch, and there's little space for the crust to grow and little light for them to photosynthesize. Another possible reason we saw greater biocrust cover was because the biocrusts in this ecosystem are either highly resistant or highly resilient to fire. So many of the organisms in biocrusts are resistant to heat stress in the form of drought tolerance. And it's possible that this heat avoidance strategy helps them survive the fire. So this is Microcoleus, which is a cyanobacteria commonly found in biocrust. And there's this layer right on top. And this is actually UV protection and prevents them from drying out during drought. Additionally, some microbes may survive fire by moving deeper into the soil because they can move. So again, this really special cyanobacteria here, Microcoleus, you can see it moving in the video, and it does the same thing in the soil. And it can move up and down in the soil surface to sort of avoid heat stress. Another possible mechanism is fast recolonization. So there are millions of microbes just floating in the air. And since these plots were located near undisturbed areas, these areas may act as a source to quickly recolonize the bare space after the fire before the plants regrew. Which leads me to my second question about the microbial community. Remember, we expected each fire to have a distinct microbial community. So first, I want to orient you to the most common microbes I found in my samples. So this is a plot of the relative abundance of the top 10 most abundant microbes from all my DNA sequences. And there are many organisms we expect to see in biocrust. So the most common is Streptomyces. This is a really common microbe in soil in general. It's filamentous, and they play a role in holding the biocrust together. Next, we have nostoc, which is a very important cyanobacteria in biocrust because it can fix atmospheric nitrogen and also photosynthesize. And it's common in biocrust around the world. It forms this like dark, gloopy slime on the soil, and we call it the dark overlord. Next is Geodermatophilus. This is an interesting genera because it's commonly found on exposed rock, and it's known for tolerating extreme conditions. Frankia is another nitrogen fixer. You may be familiar with Frankia colonizing the roots of alders and increasing nitrogen fixation. Methylobacterium is also interesting to see here because it's commonly associated with plants. It can actually use the methanol produced by stomata, and some studies have shown that they can stimulate seed germination. And finally, we have Bradyrhizobium, which is yet another nitrogen-fixing bacteria. So the top 10 genera for the control sequences look very similar to the top 10 genera overall, with Streptomyces, nostoc, and Geodermatophilus being the most common. We also have the addition of Anabaenia, which is another nitrogen-fixing cyanobacteria similar to nostoc. Then the burned sequences again have similar genera, but the relative abundance rank shifts. So Streptomyces is still the most dominant. Geodermatophilus increases in relative abundance. And nostoc, our favorite nitrogen fixer, decreases in relative abundance. So this is just a small sampling of a really massive data set, and there's a lot more to dig into. And in the future, I'll be comparing populations of key microbial groups between these treatments. So we see these slight changes in the relative abundance of certain microbial genera, but did we see a change in the overall microbial community? As my title suggests, we found yet another unexpected result. So this is an NMDS plot of all the DNA sequences. The points represent each sample, and the ovals represent the variability within the sample. So the more the ovals overlap and the closer the points are together, the more similar the communities are. So we can see that comparing all the burn sequences and the control sequences, the microbial communities are really similar. I looked a little deeper to look at the differences between fire type, and again, there's a lot of overlap between the communities. The wildfire community has greater variability, but these communities are not significantly different from each other. So it's important to note that although I did not expect to see such similarities between the communities, it suggests that biocrusts in this ecosystem are more resistant or more resilient than we thought, and this could be important for grassland resilience in the future. So in summary, we know that in this coastal grassland, that biocrusts initially increase after a fire based on data from 3 years after a prescribed burn. This might be because they are in early successional state and are eventually outcompeted by plants. We know that wildfire and prescribed fire did not change the overall biocrust microbial community, and there were no legacy effects of the fire. It could also be that this fire was low enough severity that allowed some microbes to seek refuge and survive the heat, or there may have been a microbial source nearby that allowed the microbes to swiftly recolonize after the fire. There are also differences in the relative abundance between key groups between the treatments, and this is an avenue I'm exploring now in addition to sequencing more samples to get a better idea about the population level changes between these treatments. So moving forward with this data, I'm assembling the genomes of cyanobacteria that may be unique to BioCrust or to San Clemente in general. I'm also measuring the specific ecological functions of the BioCrust in the lab. So this is a chamber with enriched nitrogen to measure nitrogen fixation. And this is a way of measuring the exopolysaccharides in the crust, which is a proxy for erosion prevention. And then what I'm most excited about is we are specifically testing BioCrust microbial resistance and resilience using a blowtorch and ceramic mugs. With that, I'd like to thank my funding sources, especially the California Native Grassland Association, my advisors at San Diego State, Dr. David Lifson, and then Valerie Ebner and Rebecca Hernandez at UC Davis, as well as my collaborators at USGS and the Soil Ecology Restoration Group, my field assistants, especially the recently graduated Dr. Aaron Oliver and Ben McKinsey, and the whole botany crew on San Clemente. And with that, I will take any questions.
All right, thank you, Brianne. I have lots of questions on my own, but let me run to the attendees very quick. There's one from Bob here. By relative abundance, do you mean found in more samples, or did you some index related to? I have a hard time with that question. Let me start over. What do you mean by relative abundance? Do you mean found in more samples, or did you find some index related to measurability in the DNA samples, such as copy numbers?
So when I get the DNA samples back, it's essentially, so I use shotgun metagenomics, which takes the DNA and cuts it into a bunch of tiny pieces, and then you get those tiny pieces back, and it identifies those pieces as individual bacterial species. And so it is essentially, I can use that to determine the copy number. And then just to account for any variability within the samples, I divided the total number of samples by the total number of microbes in general. So it's the standardized abundance, essentially. Yeah.
Thank you, Brianne. And Bob, I'm sorry if I butcher your question. I am not a scientist like this. Anyways, so I have a couple of questions. So do you think, and you may have answered that already, but the fire intensity could change things radically? And so, you know, I mean, I'm looking at this from a land management side, because obviously, you know, microbial communities in the soil are so important. But, you know, when you actively manage a grassland, like what do you do? Like, what do you avoid? So, you know, take my own example where sometimes I use pesticide, herbicide, try not to, but it does happen. And then it's grazing and burning are my main two ones. And sometimes we do fall fires to reduce the thatch. And sometimes we do spring fires, you know, late spring, early summer to get rid of some of the noxious invasive weeds also. And of course, in the fall, it's lower intensity than it is in the summertime. So like, what's your take on that? What should we try to avoid to conserve, you know, that those microbial communities?
I think having a low severity fire will definitely, it will save the species that are already present, because there's a lot of species that are already fire adapted, just living in the deeper soil layers, and then they can move up once the soil cools. In a high severity fire, the heat penetrates way deeper into the soil, and so you're decimating the microbial community at deeper depths. In terms of the timing of the burn, all of these burns occurred in this, like right before the growing season. So February-ish. Mm-hmm. But there's a previous study in New Mexico that showed when you burn BioCrust during the BioCrust growing season, it decreases their ability to fix nitrogen and photosynthesize. Or if you burn them during the dormant season, they maintain that function. And that's not something we tested here. And there's so few studies about BioCrusts or even soil microbes in general and how they respond to fire that it's definitely something we need to work on.
And then a quick question, does grazing, and maybe more specifically cattle grazing, negatively impact BioCrust?
it's very site specific. So one of my collaborators, Rebecca, she showed that in coastal sage scrub, grazing can be really detrimental to crusts because of the trampling. Although the crust and grasslands are different than the ones I've seen anywhere else. I walk over them and they seem to be fine. But again, we don't know what they're like here. But my previous work in the Great Basin and the Colorado Plateau, if there's cattle at the sites, there's generally no biocrust.
One more question. Do you think adding compost to the land would benefit biocrust?
That's really interesting. I've never thought about that.
That's your next research project.
My first instinct is, as long as there's still bare ground nearby, so because you have the compost and it decomposes and goes into the soil, and that can help the soil microbes. But the crust itself needs a bare surface.
Okay.
So if it has any mulch or any sort of micro topography, it makes it a lot harder for like those initial microbes to colonize.
Okay. That makes sense. All right, let's see. No other questions, and that's about our time. So Brianne, thank you so much. All right. Okay, so this concludes, whoa, I just did something on my computer here. Thor, you can come back anytime you want. This concludes our session. I want to thank again all of our presenters, Taraneh, Emam, Sarah Gaffney, Chris Hart, Julia Michaels, Brian Palmer. It was a great session. Thank you so much. Thor from Davis, California, right back at you.
Thanks you guys. That was a great session. Really interesting. The biocrust stuff I haven't thought much about, so that was fun to listen to. So thank you everyone for joining today, and we're going to transition into a meet and greet in a couple moments. So please hang out. I will be inviting folks who stay logged on to share your video and audio to come into the meet and greet so we could learn more from SNS Seeds, DUDEC, and HRS. And yeah, I'm happy with the great session. Julie, you have some stuff to share before we transition to meet and greet.
Yes, I do. Let's see me do this. Okay.
And everybody, this is Julie St. John, our SERCAL Administrative Director. None of this would happen without Julie. She's the heart and soul of this.
You give me too much credit. Too much credit. It's such a teamwork kind of thing. Can you hear me and see my screen?
Yes, I can.
Awesome. Okay. So I know JP already did this, but John Anderson was a part of our conferences for so long, and he made such an impact on so many people. And I'm just so glad we were able to give him a Lifetime Achievement Award. And I think he was really very pleased. It's a wood plaque. Tried to do something that would go with who he was, and it has an old-fashioned tractor on it, and it gives him the Lifetime Achievement Award. So anyway, we miss him. Okay, so let's see. Now, that's a hard segue, but I want to make sure that you know that you get a $25 discount to the Calypsy Symposium with the discount of Sim Partner 2020. Their conference, as Thor said, is coming up later in October. And please become part of the community. This is a strange thing for us to be doing, but it's necessary, and I can see that we're going to have hybrid events from now on. I mean, it's so worthwhile to have people be a part of the conference who couldn't otherwise travel. So we hope that you will continue to join us however it works for you. And please present, write articles, join the board. You know, we're all in it together. And so this is just a beautiful scene, I think from Mojave National Monument. But just thank you everyone. This has been such a crazy year, and it's been so good to have connections with all of you. You know, brief, long, whatever. It's we're a community, and community is really what we all need to continue on. You know, supporting each other and sharing information and contacts. So just a couple of housekeeping things. I think some people are not getting reminders for the sessions. I have them set up on Zoom, and I think you need to have a Zoom account, which is free. And if you all don't mind, I can send out reminders as well the day of or the day before. So just let me know if you need a reminder. I'll be more than happy to send it to you. And apologies to anyone who wasn't able to get in at first yesterday. You know, we're learning all of this as we go. We're building the airplane as we fly it. So I didn't know I needed to toggle the 500, and now I know. So anyway, thank you so much, and I look forward to the meet and greet. And please show your videos. It makes it so much more, you know, SERCAL-y. Okay, bye.
Yeah, so Julie, I will start inviting people over from Z towards A, and if you want to start at the top, start at A and bring them over. So we're promoting everyone to panelists so they could share screen and audio.
Yeah, it's so much better when we're organized like this, and you're doing one end and I'm doing the other.
Yeah. And there's a lot. We had
It is nice to see faces.
yes.
It's so weird not to be face to face. Miss you all.
And I will be able to upload this and share a link tonight, probably. There you go. We're all in.
All righty. So our meet and greet is to, hey there, Will. Spend a little time with folks from SNS Seeds and DUDEC and HRS. So Cindy,